So here we're looking at mutations, and mutations are pretty rad because they are the source of variation and all like genetic diversity. They're the ultimate source. There's other ways we can get variation in our cells and in our genetics, but mutations create variation. And so with that, we're going to look here at some categories of mutations. Now, in this little video, we're going to focus on the small scale mutations that you find in DNA, but there's also large chromosomal mutations that is more applicable to our unit in genetics or mitosis and meiosis. So let's go ahead and look at point mutations. So here, if I have a strand of DNA and we're going to do replication to make a complementary strand, you can see here with this red C that it should be a G. So that's an example of a point mutation. It often happens during replication. So here, if this strand on the bottom there is our template strand for making RNA, well, now that mutation is being transcribed into the RNA. And then when you have the codons, and each codon is coding for amino acids, here you can see how that mutation can possibly change the amino acid that the codon's coding for. Okay, so let's go ahead though and look at an example. So here, if we say that this nitrogen base is a point mutation, and then we go and transcribe RNA, you can see here how before it was alanine, well now it's coding for glycine. So this could possibly be a totally different amino acid. Okay, so let's go ahead and look at some different outcomes of point mutations. So over here on the side, you can kind of see we're going to use this as our original DNA in the template strand. So like this A would be the 3' end, the C would be the 5' end. Anyway, so then as you build your complementary, as you transcribe the RNA, here's our RNA codon, UCG. And when you use the genetic code, okay, that is going to be the code for the amino acid serine. So now sometimes you can have a mutation though, where if the DNA during replication, so if the other side of the DNA was like this, T, C, G, so this A was supposed to be a C, instead it base pairs incorrectly. Now it's an A, that's a mutation. And so now the RNA, that third base is also different. So that codon is UCU. And it still codes for serine. So when we look at our, let's go like this. So when we look at the genetic code, and you can see here with this, UCU is serine, and then UCA, UCG, etc. are all the same amino acid. And so that is called like being redundant. So there's redundancy in the genetic code, and that allows for silent mutations, where the mutation results in the same amino acid. It didn't change the phenotype at all. So some point mutations can be neutral and have no impact on that overall polypeptide. It's the same amino acid in the same codon. Then you could have what's called a missense mutation, where here the new codon codes for a different amino acid. So with this, the mutation results in a different amino acid. And if the R group of that amino acid is the same, like same chemical properties, maybe before it was a non-polar R group, and now the mutation results in a new amino acid that is also non-polar, well, it may not be that disastrous. But if it maybe mutates and now it is a negatively charged R group, that might have some impact on how that protein folds and lead to a different tertiary and quaternary structures and have an impact on the overall shape of the protein. And then our last type of point, okay, so there's some words for you. Let me move my face. So then here, this missense may affect protein folding and the final shape of the protein. And then we have what's called a nonsense mutation. And a nonsense mutation changes the codon to be a stop codon. Now, this is like not messing around. This is no nonsense. And so with this, there's no functional protein being made. So for example, if we have this mutation here that now codes like this, and we do our codons, so you can see here this third codon right here. Okay, so here we have our first two codons and amino acids. Oops. But this one, UAG, is a stop codon. There's no further protein going to be made. This says stop. So there's no functional protein. It doesn't even matter that the rest of these are okay. So stop codons can be pretty disastrous, resulting in no proteins made. So this is just a picture from Google Images that I thought was helpful. You can see here with missense, it really just depends on, here's like the original amino acid. You can see it has like similar properties. Down here it tells you if it's basic or polar. So then it could have different properties that could alter things. All right, and then we have what's called frameshift mutations. Now these ones can have pretty large impacts. So with a frameshift mutation, let's go ahead and see how maybe if you insert a nucleotide. So this is where you add an extra one. And so now here, oh goodness. Now here, when we go to transcribe it and then divide the codons, you can see by adding this extra nucleotide, everything has shifted over. So we call this a frameshift mutation. If you go ahead, you can look at the codons above in this example and compare it to the ones below. Everything has been shifted this way. So now, the first amino acid is still histidine, but the next one is now probably glutamic acid. And then as you continue to read these codons, it's a completely different protein than what we had originally. And so now, by adding one nucleotide, you've shifted everything over, and that can have huge consequences for the overall shape of the protein. Now, with frameshift mutations, it can be insertions. What I did here is we added an extra nucleotide, or you can have a deletion where you delete a nucleotide and everything shifts in the other direction. Either way, they both totally shift the reading frame of the RNA. And so the only types of insertions or deletions that aren't quite as bad is when you insert or delete 3 nucleotides together. So basically you're adding or deleting just one codon, and maybe that will be survivable if the other 300 amino acids are still the same. But yeah, when you insert or delete, it shifts everything. So frameshift mutations shift the entire reading frame, which results in a completely different amino acid sequence following everything after that insertion or deletion. So like here, this would be a deletion. So by deleting this U compared to the original, you can see now all of a sudden there's a stop codon there by shifting things over. Okay. So then this one is just showing whether to delete or insert. And yeah, just some helpful Google Images for you. Hope this was useful.